So now it's my turn, and I am going to talk about fluorescent lifetime imaging. I am glad that they give a very good introduction, so my show will be much easier and simple. But my idea is to convince you that basically, if you start from, as they say, a pulse, and you have, I don't know, a quick decay or a long decay, you can see this and this as equivalent, analysis. But I will try also to convince that if you jump into this dimension, as Enrico used to say, you need to go to the direction and you need to forget the old space. You need to move forward. You will get many, many advantages that you will not have here, and you will stress a lot, handling with your data. So, well, this is the outline, but I will jump into the why bother to measure lifetime? And I think they already said the case. Why? Why we said, well, the first claim why lifetime is something important is because we have basically very nice measurement that is independent on the independent on the concentration, right? And in some situation in cells, when we are thinking in images, distribution of a fluorescent molecule within the cell cannot be assumed as equal in different parts of the cell. And eventually you can be biased in your conclusion because, concentration is distributed different. If you are thinking in FRET, and we will see how FRET can be useful for measuring interaction in the cell, donor and acceptor can have different distribution in a cell. And then at the end of the end, you will be, concluding something that can be compromised because the situation. So having, an intrinsic measurement independent of concentration is very useful. And I said this between brackets is always, because I will challenge this and we will take advantage of when it's not independent of concentration and how we can use this situation to do some particular measurement. And also, as they've mentioned, there are many, many factors that affect the lifetime. And this, rather to be, a bad thing, is a very good thing. We have, a capability of measuring what is going on in the environment of the molecule, the fluorescent molecule, and we can measure, ions concentration, pH, hydrophobic properties. We can measure oxygen concentration because it's a very strong quencher if you have a long lifetime. You can measure molecular interaction, binding, etc. Many, many others properties of fluorophores. And this is useful because it will tell you information about this region here during the excited state, before the fluorescent comes. And usually lifetime is also nowadays a very powerful tool to multiplex, because you can have many, many fluorescent molecules, even with the same fluorescent color, but different lifetime. And you can take advantage of that. You can have, 2 or 3 similar, fluorescent molecule, like green molecule, there are many versions, red fluorescent protein, there are many versions, but they can always have different lifetime. And this is a way to expand your palette of fluorescent molecule to do research. So basically, lifetime is really a powerful tool to measure, or interrogate in vivo imaging. And the question is why not many or everyone is not doing lifetime? Well, because of course it's a technique that requires some knowledge in order to be useful. So I don't need to go in detail about these 2 schools, but now we know that there are 2 schools. One is time domain, so very short pulses, and then very quick measurements, so small windows on the picosecond time resolution, so sub-nanosecond. And then we have many, 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 many beams here. And the procedure is, well, you have the pulse, and then you tag a count. And when the photon arrives, you say, okay, that many windows, and the width of the window is, 100 picosecond. Okay, I have 2 nanoseconds of lifetime, right? And we do these processes, as they mentioned, many, many, many times, because we have the laser, pulses very, very quick. So usually in the megahertz time, so millions of pulses by second, and we collect many, many photons in order to build this histogram. And basically we rely on fitting when we have this kind of analysis. And the reason for why is because it's a probabilistic, right? We use time correlated single photon counting curve, we use fast digitizer, we can use spot arrays, we can use many other acquisition systems. And in the frequency domain, we use basically the modulation of light. And it doesn't mean that you need to modulate your light. We can use lasers. Lasers are already modulated. You have pulse or you don't have, right? And if you do the proper, mathematics, you can use the pulse of the laser for frequency domain. And it's what we will do here, and we will be doing during the practical session. So for every repetitive periodic function, which is the fundamental of frequency domain, we can do fluorescence lifetime. And basically there are a principle which is called the heterodyne principle, and we can go in detail if anyone is interested in learning, but we can use low frequency clocks mixed with high frequency, and then we can extract the lifetime from there. And this is the principle for the FLIM box. This is the card that we will use in the practical session, and is the principle for the intensified CCD and some other sensors that use frequency domain for the acquisition. Okay, why not all the people is doing lifetime? Well, the truth is, doing lifetime and analyzing lifetime is challenge. And the reason for why it's challenge is because contrary to our idyllic view of the decay, in a pixel, you have very few photons because you collect for very small amount of time. And this really small amount of time enables you to collect certain amount of photons, hundreds, but not millions. When you have to fit something with only hundreds of photons, well, you will start to scrub. You will start to having problem. And this is the reason for why when you have many more components, let's say 2, 3, or you have reaction like they stay, you have thread, you have many other situations, well, this can be challenged. And the other important point is, well, when you have to do fitting takes time, and it takes computational calculation. So this is another drawback of the fitting. And at the end of the day, basically, we end up having a technique which requires a lot of expertise. So you need to become, in a way or another, an expert in order to do that. And this is something that compromises, of course, the use of the technique, because not all the people, first of all, want to be an expert, and they want to just obtain the information, right? And also, it requires, certain amount of time of your life in order to make it work. Okay, so the last thing that I want to mention about why not everyone is doing fluorescence lifetime imaging is because there are not that many commercial microscopes that you can buy, with a key in the hand, and you push the button, and you get the decay. So far now we have the Leica SP8 with the Falcon Stellaris model, which is a commercial microscope that you can order, and they will set it up in your lab, and you will have lifetime without basically any trouble. So it's something that is like a confocal microscope. And there are other companies who are also pushing in having, more commercial version, but most of the time they are upgrades of existing technology, or upgrade to a technology that you buy and you combine, a science microscope with the Pico Quant or the Becker and Hickel or something like that. So can we do something for not expert? And I like this question. There is a chance. Well, eventually, we need to jump into new space. And this new space, as Enrico like to say, is the phasors. If we move from the Cartesian space, intensity, decay, to another space, which everything is possible because we have other rules, other logics, well, maybe we can succeed. So Enrico already mentioned this. The mathematics for the phasor was introduced by Gregorio Weber, in the early 80s, but was not used until, Enrico, Dave, Bob Hall introduced the plot, this phasor, with this hemicircle that we call universal circle, and it's universal because it contains all the single exponential decays. And since Weber notation, we have, all the mathematics for the frequency domain and time domain. And basically, this produce a transformation of the data into this new plot, who has some advantages. Well, there is no knowledge a priori that you will need in order to understand what is your system or what your system is telling you. And we will learn why. Why? This is magic? No, it's not magic. There are few rules that you need to learn. These rules are very simple. And the most important point, you don't need to fit. So by removing the fitting, you remove from your path one of the most important constraints in order to use FLIM, which is the knowledge of the model, physical model, to use for the fitting. And I want to point to this paper that I just published a couple of months ago, which is more a perspective and a view of the future of how Phasor plot can be, the tool in order to democratize the use and the access to FLIM, because it's not only true that it's useful, the fluorescent lifetime, but it's also useful for other dimension. And in the afternoon, I will talk about, how we can use Phasor also for hyperspectral imaging. So basically, Phasor is a tool that you can use in any periodical function. So if you collect your image with any periodical function, you can do Phasor transformation. And can be true for polarization, can be true for FLIM, can be true for spectra, etc. Okay, but what we mean or how we do the Phasor transformation? Well, we have a file. When we open a file, you will see in each of the pixels a decay, right? So basically, our pixel, instead of have a grayscale, it will have a third dimension, which is our decay. And we will do this mathematic. The computer will do this mathematic for us. And for each pixel, it will have many millions in an image that we acquire nowadays. It will build, a cluster in the Phasor. And depending on where the cluster is in the Phasor, we will understand what is going on in our sample. But how is that the calculation? Well, if we have a fast decay and we multiply for this cosine or a sine function, like this, this is this cosine or sine function, you will have, a modification, of this decay. And if you integrate this, if you sum all this point, which is the numerator here, and you divide by the integral of this, you will have two coordinates. You will have g, which is the x, and you will have y, which is the s. And this will be in a point. If this is on top of here, it means something. If this is inside here, it means something else. And there is something very interesting of the Phasor. If you have a short lifetime or a long lifetime, it will pop up in the Phasor in different regions. So short lifetime will be always close to 1, 0, and longer lifetime will be towards 2, 0, 0. So all the way down to counterclockwise, right, to 0, 0 position. So by looking at the Phasor, you already will know many things of the decay without even know what is the sample you have or what are the fluorophore you have in the sample. And this is powerful. So as they say, well, this universal cycle contains all the single exponential decay. And if we have two single exponential decay blend in a certain amount, well, they will show up at the linear combination. And the counterpart of the distance between this to this is the fraction of the component 1, and the other way around is for the component 2. So using very simple geometrical algebra, we can quantify, not only see, but quantify what are the amount of one fluorophore over the other. And in a graphical approach, which is very simple. But we can also learn that if we have something outside the universal cycle, as they mentioned, it's because something else happened before the fluorophore emit the lifetime. And this is when we have reaction in the cyclic state, as they say. If we look at the FRET, in the FRET, we look at the acceptor. The acceptor will emit photons, but it will not emit any photon before the energy transfer from the donor. And these two processes take time. This time is nanosecond time scale. And this is the reason for why it pulled out the information outside the universal circle, right? But there is one very important properties of the Phasor that also they pointed out, very important today early in the presentation, and this was the big, idea of Enrico of using Phasor, which is for images, we need to know where things happen. And if we do the transformation, we have a fluorescent lifetime. This is an average intensity, but we transformate data and we end up having something in the Phasor. We can go to Phasor and say, where data that I found here is in my original image. So I can use this reciprocity between the Phasor transformation and the image back and forth in order to learn what is going on pixel by pixel in my sample. And this is really powerful thing because otherwise you need to do fitting. And the fitting can work in one pixel but not in another, etc. So this reciprocity principle goes in this direction, but goes also in the other direction. So if I decide to do very small region of interest here in the cell, you will have scatters in the scatter position in the phase, which tell you how much heterogeneity you have in the sample. So this is really useful, and we will take advantage of this during the practical session and the analysis, because we will realize how much we can extract from our sample. So these are the properties of the Phasor. This is the language that we need to learn. After we learn that every single exponential decay is in the universal circle, that the combination between 2 points give you, how much you have of one and another, that the position outside universal cycle is because something else was happening before fluorescence, we are done. We learn the new language in order to understand how Phasors are telling you about the fluorescent decay. So, but Phasor, it has also very unique opportunity, because most of the time we have very few photons, and the precision in the measurement is always relies on the inverse of the square root of the number of photons. If you have few counts, your distribution in the Phasor will be broad. If you have a lot of counts, you will be very sharp. But with the Phasor, we can use filtering in the Phasor space, not in the Cartesian space. We will blur your image. And using this filter in the Phasor space will enable you to reduce the requirement of photon, so your photon budget, and you will still have the precision to measure the lifetime with high accuracy. And this is an example we did. We have 500 counts, and you see you have very well-defined points to define, all the linear combination. This is a single exponential decay, another single exponential decay here, another here. And we did mixture of the many different conditions, and we can, using the geometrical calculation, we can know how much we have of each in this point, or in this point, or in this point. If you have 50 count, well, you start to struggle, right? You start to see that the cloud is a little bit noisier, more broad. But we can apply the filter. And if I apply, a filter of a kernel of 3 by 3, so this means if we have a pixel in here, we will see what are the position around, and we will correct if this pixel here has an outlier, whatever measure which was, I don't know, far away for whatever reason, noise, etc. And if we do so, you see that you already improved. But if you use a 5 by 5, you improve a lot. You improve in a way that you are comparable with 500 counts, only collecting, 50. So this is another very powerful use of the Phasors. And it's hard to do this when you have to, filter, decays. But with the Phasor, we can obtain quantitative information, and we can obtain quantitative information of our lifetime even when we don't know the lifetime that we have in the system. If we have, our system calibrated, and we calculate, let me go back to here. We calculate here, not the first harmonic, but the second harmonic. So a second frequency, so 2 periods of cosine or sine, we will get another set of data that will come up in the phasor in a different position. So let me show here. So we will have, this is the first harmonic, and if you twice the frequency, you go to second harmonic. If you do the math, there is only one combination that match, the fraction of component 1, the fraction of component 2, the lifetime of the component 1 and the lifetime of component 2 in the two frequencies, in the frequency of 80, the frequency of 160. And, the software can do this interactively and can give you information about what are the lifetime, if you know there are two lifetime, and, what are the fraction of each, just by fixing this point in the calculation. Using the phasor, you can solve 3 components using 1 harmonic. And this is because you have this mathematics. You have the sum of all the G's, the sum of all the S, and their prefactors. And then you have the sum of all the prefactors. So the sum of all the prefactors is 1. So you have 3 equations, 3 unknowns, so you can solve this equation. You cannot have 4 with the first harmonic. But you can go again to another harmonic and get another set of G and S for the second harmonic, so you can go to 5th. And this is a method that probably today in the afternoon, Alex Palmigiana will explain, that is very powerful. You can solve 4, 5 harmonics, component using the harmonic information. And even more, they manage to do this in a blind way. They can obtain information up to 4 components without knowing what are the lifetime of each component or even the fraction of each component. And this will be something that he will introduce. So I will not spend my time talking about fluorescent rational energy transfer, but basically what we want to know is if 2 molecules are interacting. And nowadays we can, include in DNA, in plasmid, a fluorescent a protein of interest with a fluorescent protein or other kind of labels. And if we want to know if a molecule can interact with some other, you can see by these processes of Forrester energy transfer the proximity of these two. And this is because, the FRET is very, very or strong dependent on the distance of the two fluorophores. It's also dependent on the alignment, so how the dipole are oriented. And this is one of the things that I always like to highlight. A positive result of FRET is very, very nice. The negative doesn't tell you much, because can be that you don't have FRET, not because your molecules are not interacting, it's because they are not well aligned. And this is something that can compromise the use of FRET as a fair choice if you want to use this tool for molecular interaction in the cell. But the other requirement is that the donor and receptor has emission of the donor should be an spectra overlapping of the acceptor excitation spectra. But if you have all these conditions, you can measure, energy transfer with a range of distance which is very, very close. So you can measure, distance of the Armstrong scale, so few nanometers. And this is a tool that exploit its use when we can build biosensors to measure, enzyme activities within the cell. And I will give you an example. So we can measure FRET efficiency using ratios between, intensity of the donor in the presence of the acceptor, or we can measure lifetime. And I will try to convince how important it is to use lifetime and how powerful is even more when you do have phasors, because with the lifetime alone, eventually you can also be screwed. So they introduce, this, that if you have a lifetime here, right, from your donor, when we measure FRET in FLIM condition, we only take care of the donor. We don't care about the acceptor, right? So this is an advantage. You don't need to do all the calibration for the acceptor, because the only thing that is matter for us is that the donor will quench and the lifetime will go short. And we learned from the phasor plot that when we have shorter lifetime, where will be the position? Well, toward to the 1, 0, right? And this is true for single exponential decay, but it's also true for not single exponential decay, for a mixed fluorophore. That can happen. There are fluorescent proteins that are not single exponential decay, but still the quenching will go toward here. But the Phasor plot give you also another advantage. In the cell, and in the condition that we always do experiment, we don't want to have the cell full of fluorescence, because otherwise you will modify the function of your cell. You will affect, the metabolism, etc., etc. We always like to have the cell who express your fluorescent molecule, but not in that high amount, right? So you will have autofluorescence. And if you will have autofluorescence, this is another component. But this is not a problem, because in the Phasor we will include this other component, and the Phasor rules will tell us how to solve it. So we learned that the quenching will go in this direction, and if we have another component, autofluorescent, it will be the combination of the point here, which is some amount of quenching, so fretting, and the autofluorescent. So the point will be at some point here, and then if it fret more, it will be here, or here, and here. And you see that this trajectory is some sort of a curve. And you may ask why? Why is a curve? Well, as increasing the efficiency of fret, so more quenching, you will reduce the quantum yield of the donor. But the autofluorescent remain the same. So the linear combination will pull your data to the autofluorescent. So you see that just using word, I can explain this very complex photophysical processes, and you can use the rules of the Phasor to quantify this and quantify the quantum efficiency, even in the situation where you have strong autofluorescence. And this is true also for multi-components. So with the SIMFCS software, which is the software that Enrico developed for all of us, we can measure the quantum efficiency just dragging these bars here and setting up the donor, setting up the autofluorescent, setting up the amount of donor that can be quenched, which is this black here, and this will tell you, okay, with this condition, you have 0.2082 fret efficiency in this cloud of data from your cell. So it's really, really, really easy and power. And let me show you how power can be and how complicated it can be even to fret a measurement in cells when you are acquiring only lifetime and you are relying on fitting. So this is a case of two molecules that interact. They form dimers, and they have labeled this this protein with EGFP of the MR red fluorescent protein. And then you have, certain, like 1,000 of count per pixel, which is reasonable for FLIM. And you see, well, this is the average intensity. When we do the fluorescent decay and the fitting, you will see that there is a distribution here of lifetime over counts. And you have a long lifetime, which you assume is the donor. And you have a short lifetime distribution here, which we assume is the donor in the presence of the acceptor, so fret. If we color this based on fluorescent lifetime, you see that the more cyan here is distributed over here, and this means that this is the distribution of where your molecule is doing dimers and fret. But this molecule, I didn't say here, it aggregates at the cell junctions, so where the cell tight. And this is very weird because I will assume that it's not well distributed in the whole cell. Well, let me show you what happened. Well, we were not considered the autofluorescent in that situation. And let me show you how easy it is to solve this with Phasor. If we put here, this is the donor alone, and you see the donor alone highlight all the cell. When we look at autofluorescent, you see that here is the position for autofluorescent. There are autofluorescent somewhere here. When we go to the trajectory that predict the fret, like here, you highlight only regions where you have the gap junction, when you have the cell-to-cell interaction. And why it is totally simple to see that, we were missing, where the location was, because we were looking only at the quenching, but not the quenching in the presence of autofluorescent. So the Phasor solved the problem very easy. This is for molecular interaction. For most of the application nowadays, for biosensor, and biosensor can be built in a way that you can, use part of an enzyme and see how this enzyme is, changing in the cell and distributing the cell. And you can put, optimize the fluorescent protein for the overlapping. And in that case, this is an example that I work. There are one biosensor, which is a biosensor for this enzyme. And using the Phasor, you can see we can follow over time. This is the cell doing, ruffling, micropinosome. This is vivo imaging. And you see at some point here, there are like some macropinosomes getting in the cell. And this disappear over time because, well, the cell rack does the work and then goes away. And you can quantify this over time. In this case, where we were interested in knowing, the effect of a drug and how the drug affect the activity of the drug one. And you can do, quantification, and you can do, illustration of threading processes very easily using the Phasor approach. But perhaps the most important and powerful use of the Phasor nowadays is the application on the autofluorescent and the use for the metabolism. And autofluorescent is something that we learned from very early days in the 60s from Victor Chance. That is a powerful tool to measure, the metabolism of cell, because it relies on the NADH and FID. So the DPNH here is the way that they used to call the NADH early on. And Britton Chance shows that, if you change the amount of oxygen, you can see changes in the intensity of the autofluorescent, and this was correlated with the metabolism. Nowadays, we can use two-photon excitation. The big problem here was you have to use UV excitation because NADH is excited at 360, 370. So this is very bad for in vivo imaging. But now with two-photon excitation, yesterday we learned that using two-photon excitation of the half of the energy, we can excite the same molecule. So instead of using 360, we can use 740 excitation, and we can strong excite NADH. And this is interesting because NADH, as most of you know, is related to the, is produced during the T-carboxylic acid cycle, and it's used in the mitochondria to produce the energy. So NADH can be in the cell as a bound or as a free version. And lucky to us, the change between bound and free condition, it changed the lifetime. And here it is, some values about the change in the lifetime. In free is 0.4, while depending on which, enzyme NADH is bound, it will go from, few nanoseconds to 5-6 nanoseconds. For the LADH, in certain condition is 3.4, and I will show you later. And also the FID changed lifetime, and it was introduced early on, this radiometric measurement of intensity of FID over intensity of NADH as an optical redox ratio. But now we can use lifetime. And the thing of lifetime is, as I mentioned before, the version of free and bound of NADH changed the lifetime. And this is a plot. This is a plot of the decay of the fluorescent of free NADH. And as soon as we increase the bound fraction by adding, more and more enzyme, this is bound to LADH, you see that the lifetime go up. And we can use this change of NADH bound and free as a ruler for the metabolism because it correlates very well with the ratio of NAD plus NADH. And this was, corroborated or, compare the optical ratio with the oximetry measurement of the metabolism, like, seahorse or Ouroboros and other things that rely on the oxygen consumption. And you see that they both follow the same path when you stress the mitochondria with different, drugs. So as I mentioned, we can build in the Phasor plot a ruler to measure the metabolism. And, if we do, a titration of the binding of NADH to an enzyme, let's say lactato dehydrogenase, we will have a linear combination from very short lifetime, 0.4, to long lifetime, 3.4 nanosecond. And this ruler is now a metabolic trajectory, we call. So if we have pixels along here, which are more or less the location of the pixel that we will get from cells, we will be able to measure how much it changes the metabolism. And what we learn about these changes in the lifetime is that if the lifetime is short, so if you have higher amount of free NADH, is because you have a more glycolytic condition. And this is shown here, where you have glycolytic condition or you have phosphorylative condition. When we have longer lifetime, so we have more bound, NADH, is because the cell is preferred to go to the phosphorylative oxidation of the glucose. So using just this ruler of the Phasor of free and bound, we can build this metabolism. And this is really, really useful now for diagnostic, for understanding, many effect of drugs in cell, etc., many other things. So I will move quick because I am a little delayed. But to do this magic, it requires some proper condition for Phasor transformation. And one of the things that we were discussing before is how much comparable, time domain and frequency domain they are. Well, you can use, average lifetime, but average lifetime doesn't tell you more about the free and bound. And the problem with the time domain is if you collect data and you cut data because, you conceive the measurement from the point of the pulse to the arrival of photon, but not in the period of the laser, you chop the data. And if you chop the data, when you transform the data in the Phasor, you can screw up the measurement because you can overestimate or underestimate the amount of free or bound, particularly bound because it's the long lifetime, right? So how we deal with that? Well, in the high frequency lasers, like 80 megahertz titanium sapphire is the one that we will use here, your frequency is fixed. So it's 80 megahertz, which means that you have a window of 12.5 nanosecond. So you have to collect photon in this 12.5 nanosecond. If the photon, this does not hit the detector at some point, it will come up another burst of the laser, and that's it. You are done. So how we acquire the data in the car, as you were asking before, in the time correlated photon counting, we have the pulse and then we decay, and we match this with the period, but usually discard part of the decay. So you don't have all this part of the decay here. Well, we managed to solve this problem using time correlated photon counting, and we did it in this way. You collect 2 periods, right? And if you collect 2 periods, this is just changing how the car acquires the data. You can set, a period between 2 pulses, and you will have a full decay. And in that case, I will show you in a minute that the transformation of the Phasor work very, very good. As good as the frequency domain. Because in the frequency domain, the only thing that matters for us is the period. Okay? So just to show you here, this is the titration of NADH free to bound. And you see we prepare solution 4.4% of NADH bound, and then 44, and then 100%, and they are all in the line. When in the frequency domain, in the time domain, you see that if you do this trick that we develop, is easy, and you get the same result. When you do the classical acquisition, you see that all the points are around, and you can screw up your measurement. So, in complete period, Phasor transformation is done. You can do the Phasor transformation, but you cannot quantify. You cannot not use all these rules. And how much we can influence? Well, we can influence a lot. In imaging, let's say that these cells are the same. this was acquired one after another. We have a microscope in the LFD, Enrico's lab, where we have time domain, frequency domain, and it was one acquired after another. And you see that in the situation where we make this little correction, it was comparable. But when you have, the condition that the cards usually acquire is totally out, and you can see how much bias you can be. So the next application that I want to mention is about Phasor plot to use absolute concentration of molecule. This is set for the NADH, but can be used for any other fluorescent molecule. And I mentioned at the beginning of the talk that fluorescence is independent of the lifetime. And this is true, but if you have 2 fluorophore, the position in the phasor will be the ratio between the amount of 2 fluorophore. It's independent of lifetime, but now we can use this linear combination rule in order to quantify how much I have of a fluorescent molecule that I don't know. And how we can do that? Well, this was a paper that we did with Enrico, with Michelle, and Michelle's student, Ningma, where we light our sample with unmodulated light. And unmodulated light is infinite lifetime for our measurement, which was basically to put a flashlight on the microscope, more or less. And then when we dope our measurement with this flashlight, where will be the position of a lifetime which is infinite? The position will be in 0, 0, because this is the longer lifetime that we can go. So if we know that we have a lifetime here, whatever the other lifetime that we have in the phasor, if we have both, will be in the linear combination between these 2 points. And if we do the proper calibration, let's say we measure a solution with certain amount of NADH in this case, and we light our flashlight, we will have some, movement of the trajectory as shown here. And if we increase MOS, we have more. So if we have different amount of the fluorophore in different pixels, they will show up in this linear combination. And the calibration will enable us to give you the concentration. So this is the situation. We have cells, and the cells, the NADH will be between free and bound NADH. But when we put the light, this trajectory will show up here. And these pixels that are back is because they are high concentrated, and these pixels that are close to this point is because they are weak in fluorescence. So using this ruler, now we can quantify the amount of free and bound NADH in absolute term. And we can build an image which is the quantification of the NADH concentration in the cell. But I insist, this can be done with any fluorophore, any fluorescent molecule that you can have in the sample. So to finalize, I will just want to show a couple of more applications. This was also developed in the LFD, run originally by Ruth Sadata, and it was, the discovery of a fluorescent species that we didn't know where they were, from where they come from. But what we observed was when they were looking at cells that were, eating some lipids, the metabolism measurement start to appear this, group of pixels here that point to certain other region in the phasor. And this region is here, which is 7.9 nanosecond. And they discovered that this was due to lipids. This happened within the lipid droplets. So they discovered this is because of oxidation of lipid, and they call long lifetime species. And this was only possible because the phasors, because we didn't assume a model, and the transformation of the phasor show up this tail pointing to this region in the phasor, and it was not evident for any other time domain guy because they would not include this situation in the lifetime, right? And now, Sum and Ranjit was used this for, quantifying, in liver of mice with different diets, how much this species can have. So to finish, I want to say some words about the phasor. Well, there are many obstacles about in FLIM but all of them can be removed using the phasors. The phasors are absolutely quantitative approach, right? It's a global analysis, so we can analyze many images in parallel. So we can put all our samples in one condition, another condition. We can use, segmentation to, and we will see in the practical session, to collect information for different regions within a cell or different group of cells, etc. The interpretation is really quite straightforward. My talk was very easy to follow because there's no really big requirement. You need to learn 2 or 3, new words to understand how Phasor works, and it does work in the time domain, in the frequency domain. So, as Enrico like to say, well, the Phasor is a radical, a different way to analyze lifetime, and I welcome all of you to the Phasor space. Don't look back. When you jump into the Phasor space, you cannot come back to the time domain anymore. It's not true, but anyway. And I want to mention that these are the 4 instruments we have with FLIM capabilities in Uruguay. As I mentioned yesterday, these 2, the inverted microscope in the hospital, the upright microscope here. And then we have another project, which is a fiber optic to measure metabolism. It's a metabolism profiler, we call, with fiber optics. So the idea is the fiber optic will be inside an animal or inside an organ. And we receive, is in the hospital, in boxes, a donated instrument from Claire Brown, McGill University, where we have also a Peacock 1 system to measure that. So thank you very much. I want to point that we will learn more about the Phasor Pi, which is the new library to do phasor analysis on hyperspectral imaging and spectral phasor. And Bruno is also developing this Napari plugin that we will teach more in the practical session. And I want to big thank Enrico, because without him, we wouldn't have, all these phasor and all these ideas. And the first time that I hear some of these methods like absolute concentration and some others that we will learn along the cruise, I was, over my mind. I couldn't imagine how this can happen, but well, Enrico always managed to have great ideas, and also to do, all the software and all the requirement in order to get the information that we need. So thank you very much, and I am happy to take questions.